Back for a video on hemoglobin and three key characteristics about it. We're going to talk about the cooperativity of oxygen binding to hemoglobin. We're going to talk about hemoglobin affinity, and then we're going to talk about blood carrying capacity, also related to the amount of hemoglobin. All right, so we've got our oxygen dissociation curve. I've already sketched it out here. Notice our y-axis is percent saturation of hemoglobin with oxygen. There is a separate axis label that we can use to assess capacity, right? Capacity being the amount that we can carry again. So we're going to use this one first. And remember, at 100% saturation, that means all four of the oxygen binding sites on hemoglobin are bound on essentially all of the hemoglobin molecules in the sample that we're testing. Remember, this is an experimentally derived graph. We use it to explain the physiology, but it's not truly physiological. We don't slide up and down the graph. Okay, so and again, any individual hemoglobin molecule can only be 100%, 75%, 50%, 25%, or 0% saturated. If we average a population of hemoglobin molecules, then we can end up with values in between, because some of those hemoglobin molecules might be, in here, some are 25 and some are 50, and so depending upon the proportion of those two, you end up with a saturation value that's between those sort of whole number percents, when it's 1, 2, 3, or 4 hemoglobins, 0, 1, 2, 3, or 4 hemoglobin, oxygens bound. Okay, so first thing to point out before we start looking at the curve itself, is that at a PO2 of 100 in the lungs, again, maintained by ventilation, or tidal ventilation, right, explained by the, Boyle's law and things like that, we're going to keep the alveolar PO2 at about 100 millimeters of mercury. PO2 down here, it's in millimeters, always in millimeters of mercury. And one thing to point about that is that value of 100 will stay true even if we start to remove oxygen from the lung, the respiratory membrane surface, more rapidly during exercise with increased cardiac output. And that's true because ventilation will go up in sync with the increase in cardiac output. So in other words, if we're going to remove oxygen more rapidly, we have to add oxygen more rapidly, right? So ventilation and perfusion, blood flow, need to go up and down together to maintain this 100 millimeters of mercury here. Now, at the systemic tissues, the PO2 is more variable, right? Here, unless you're at high altitude or you have a respiratory disease, it's going to be 100. Some people, I might have called it 105, just rounding it off to a nice round number. The tissues are much more variable based upon whether or not the tissue PO2 is more variable based upon whether the tissue is at rest, in which case it will have a PO2 of about 40. Squeeze that in here. Or if the tissue becomes much more active, and sort of like, and I don't want to put this as the lower limit, but this is sort of the normal range of tissue PO2, and an active tissue drops down to about 20. Okay? So, and again, so we'll talk about whether or not we're resting or the tissue is active. And I usually use exercise as an example, so we're thinking about skeletal muscle. And the best way to think about that is it's because the reason to think about skeletal muscle is it has the most dynamic PO2. So we see the biggest changes on the curve up here when we look at skeletal muscle. If you ever look at brain PO2, you could almost probably pick a number here, right? Unless something really bizarre is happening or you're at very high altitude. Brain PO2 is going to probably sit around 40. And we would only really have to ever look at the curve at one place. You could increase blood flow to drop off more oxygen, but the tissue PO2 would be more stable. Okay, so again, we've got a big chunk of our body mass that has a much more variable, and so we'll assess a couple different tissue PO2s. All right, so basically what we need to start talking about is these three characteristics, right? So we're going to start with cooperativity. So cooperativity is indicated by the fact that this line is not linear. It is a sigmoidal shape, and that's because binding of oxygen to one of the four binding sites actually alters the binding at the other binding sites, right? So in other words, when one oxygen comes on, it makes the next oxygen come on more rapidly, right? Meaning a smaller change in PO2, the line got steeper. And that's cooperativity, right? It is a form of allosteric modulation. We call it homospecific because it's oxygen binding affecting oxygen binding. But basically, the whole point of it is that it makes the curve steep, and you can see that the steep part of the curve actually sits on top of the tissue PO2. So what this means is that small changes in PO2 result in large changes, right, in percent saturation. Percent saturation in this scenario with the tissues is basically telling you how much is being delivered, right? So again, we can look at the percent saturation. If we want to say the hemoglobin of the tissues was 50% saturated, if we just pick a number here, right, and go over, right? We're 50% saturated the tissues. Well, that means we've unloaded 50%, right? 50% remains on the hemoglobin, saturating the hemoglobin, but only half of the sites are saturated, and so the other half has been unloaded, right? And so what we need to do is assess how far left here we shift, right? Because the farther left we shift, the farther our percent saturation falls, and the more we will unload, right? So again, it's important to keep that in mind, is that 25% saturated means 75% delivered, okay? Now, over at the lungs, right, we're always going to end up with 100% saturation outside of respiratory diseases and high altitude, things like that. So, but that sigmoidal shape does describe the cooperativity of the oxygen binding sites with the other oxygen binding sites. Okay. Now the next thing that happens, and actually before we go there, right, let's pick a scenario here where we change the tissue PO2. So I think in class what I did is I picked the resting PO2, and so if we go up from there, right, we read over, right, we get a spot on here where we are 75% saturated, right? So if you're 75% saturated, again, that means you've unloaded 25%, or 1 out of the 4 oxygens on average that most hemoglobins are carrying. So at rest, we're basically doing this, right? We're going to the lungs, 100% saturated. We go to the tissues, we go down to 75% saturated, drop off 1. Back to the lungs, go back to 4. Tissues 3, lungs 4, tissues 3, right? So we're just sort of going back and forth, loading and unloading a single oxygen off of that hemoglobin. The other 3 are basically going round and round and round, and we call that the venous O2 reserve. Because if you think about it, the 75% saturated is what leaves the tissue. We're dropping off the 25%, and we're leaving the 75% to return to the heart and then the pulmonary circulation, right? And so it's called venous O2 reserve because it's the amount of oxygen that doesn't get unloaded, remains in the venous blood, and would come around again in the arterial blood in the next circuit. So it's reserved because if we change PO2, as we'll see in a second here, we can unload it and enhance our oxygen delivery simply by dropping our PO2. Right? And so let's change our tissue to the active state. Right? And so now you can see we're dropping to about 25% saturated. Let me extend this line over. Right? And now we're going from lungs to tissues like this, where we've tripled our delivery, right? Because we were delivering 25% or 1. Now we're dropping to 25% saturation. Therefore, we're desaturating by 75%, so we're unloading 3, right? So we tripled our O2 delivery simply by dropping our tissue PO2, right? So we're going, now the oxygens are going, 1 bound, 4 bound, 1 bound, 4 bound, instead of 3, 4, 3, 4, right, the way we were before. Now, it can get even better than that because, number 1, we could have each hemoglobin unloading 3 instead of 1 when we're active, but we can also, and we do, speed up the blood flow. So we could have more blood passing through the tissue, unloading 3/4 instead of 1/4, right? So again, when we looked at VO2 in lecture, VO2 is a measure of the oxygen consumption, and it's based upon how much you're delivering, 1 or 3, or even more than 3 on average, and how fast the blood's flowing, right? So VO2 actually equals cardiac output times the amount of oxygen that's being delivered per hemoglobin, right? So that's our change in cooperativity, and then our change in PO2, that because we have good cooperativity, is a very large change in percent saturation with simply just a drop in oxygen at the level of the tissue. Now, having said that, we can make it even better, right? And the way we make it even better is by reducing the affinity. So characteristic number 2 we want to focus on. Changing affinity means that we bind something else to the hemoglobin molecule, and it modifies the likelihood that hemoglobin stays bound to the oxygen. Okay? So, if, for instance, at the tissues, one of the things that happens at tissues is this is where we're producing our carbon dioxide, right? We're getting rid of it over here at the lungs. We're producing the carbon dioxide here. Well, that carbon dioxide at the tissues binds to the hemoglobin molecule. In addition, carbon dioxide, because of its combination with water in the carbonic acid equation, results in the production of free protons, H+. That also binds to hemoglobin, altering its affinity for oxygen. So instead of, right, so if we were on this curve, right, at the lungs, we really wouldn't be on this curve at the tissues because the CO2 and the H+ are different, right? In fact, even the temperature could be different, right? Because working, especially muscle tissue, generates heat, which also changes the affinity. It's not a molecule that binds, but it does change its affinity in a different fashion. So again, you wouldn't be on this curve. You'd be on this curve at the lungs, but you wouldn't be on the same curve at the tissues because this curve was generated with a hemoglobin sample where all we did was change PO2. That's not the only thing that changes, right? So at the tissue, I just said the PO2 changes, but so does the H+, so does the CO2, so does the temperature. So all those things, increased H+, increased CO2, increased temperature, cause a decrease in affinity. And so really, you'd be on a curve that sits more shifted to the right. And I can draw that all the way to the top, but again, you wouldn't be on this curve at the top because the pH and H+ and everything will shift to the opposite direction. I'll explain that in a second, right? But what this means now, if we just, and I drew this arbitrarily shifted by a certain amount, but if you check it out, right, if our active tissues, right, at our active tissues, we're producing more CO2, more H+, so you're more shifted than normal when you're active than when you're at rest. And so this would be the red curve would be the active curve. But check it out. So now at the tissues, instead of becoming 25% saturated and unloading 3, you become, call it half of that, 12-ish, 12.5 if you want to be exact, 12.5% saturated, half of the 25, right? So in other words, you've increased your unloading again. Right now, it's not a huge difference, but every little bit you can unload when the tissue becomes active, helps that tissue remain aerobic, especially during exercise, right? Now, again, when we get to the, when this blood comes back from this active tissue and goes to the lung, well, what does the lung do? Well, the lung gets rid of carbon dioxide. We just said the carbon dioxide is binding to the hemoglobin here and shifting to the right. So we get rid of the carbon dioxide, we shift left, right? And so we shift back to the white curve. Because we got rid of the carbon dioxide, the carbonic acid equation shifts back towards the CO2, and the H+ falls down. Okay, well, that was another reason we had shifted right. So again, we're undoing everything that happened here that caused the right shift up here, right? Even temperature, right? We lose a lot of some of our body heat during exercise through ventilation, right? The warm blood is coming to the lungs. A lot of it, all your cardiac output goes to the lungs, right? And so you're dissipating that heat into the air that you're exhaling, right? Your warm breath gets exhaled and you lose body heat. So that's going to actually help increase the affinity, which is exactly what you want. You want high affinity and high percent saturation at the lungs. You want lower affinity at the tissues, and you want it to be especially low when the tissues are active, right? Just for the purpose of naming things, the H plus CO2 effect on affinity is called the Bohr effect. The temperature effect doesn't have a name. There is one other molecule that we have to point out, another modulator of affinity, right? So this is caused by this shifted curve. It's caused by H plus CO2, and again, increased H plus, increased CO2, increased temperature. These two were the Bohr effect, right? And the fourth item on our list is 2,3-BPG, biphosphoglycerate. Some people call it DPG for di instead of bi, same thing. This is a molecule produced in glycolysis in a metabolic pathway of ATP production, and it actually causes the same shift, the same rightward shift. Right, this curve is rightward shifted. Because it has a lower percent saturation, it has a lower affinity. And keep in mind, undoing these things, having these things shift in the opposite direction, shifts you left, right? Which is, again, this is exactly what happened at the lung, for the first three at least. This one is stimulated. It's kind of like you always have this stuff, and it's always shifting you right. It's kind of giving you the curve, your normal curve. Now, the amount of it can increase, just like these. This is always present, always present. There's always a temperature, right? There's always some thermal energy. Question is it higher than normal, or is it normal? How much of a shift here in the tissues do we need? The BPG is stimulated by hypoxia. All right, well, that makes perfect sense, because if you're hypoxic, you want to unload more, you drop affinity, perfect, helps solve the problem. The harder one to explain or understand is it's also stimulated by the increase in pH, right? So remember, these are stimulants, or a plus sign here for stimulating BPG. Well, an increase in pH would actually shift us left, right? And the BPG compensates for that by shifting us right. So kind of like, it's like you shifted left, you shifted right, you end up kind of like where you need to be, right in the middle. When does pH go up? Well, some respiratory diseases. Another scenario is at altitude when you hyperventilate. So if you hyperventilate, you blow off a lot of extra CO2, right? You exhale to get rid of CO2, you hyperventilate, you get rid of more CO2. That affects the carbonic acid equation. Remove this, right? Take the CO2, have that fall in that equation, and the equation gets pulled toward the CO2 that's disappearing, right, to produce more of it. And what that means is that the H plus falls, right? If H plus falls, that's indicated by an increase in pH. So at a high altitude, you hyperventilate, CO2 falls, and your pH rises, right? At high altitude, when you're potentially hypoxic because of the lower partial pressures of gases, right? Lower atmospheric pressure, and then because of that lower amount of oxygen in air. Number one, you're hypoxic, so that's helping you, still unload more. But you're hyperventilated, which actually hurts you at the tissues, right? You're hypoxic, and shifting left would not be good. And so the 2,3-DPG, or BPG, helps shift us back and maintain normal affinity for unloading at the tissues, even when we hyperventilate. Okay, so those are our allosteric modulators. Temperature is not an allosteric modulator. That's not a molecule that binds it, but 1, 2, 3 allosteric modulators that affect affinity of hemoglobin, right? Causing the curve to shift right, or again, if we remove them, shift left. The last quality parameter for hemoglobin we want to assess is capacity. Now, capacity, we can't use this axis, this label on the axis. Basically because all curves would end up with enough oxygen, would end up at 100% saturation. And we need that to separate them out. We need to use a different label here. So I have to get rid of my percents. And what we're going to use, I'm going to get rid of my curves. I can get rid of all this stuff for a minute. Leave this one. We're going to use O2 content now. Like basically the O2 content capacity. How much oxygen can the blood carry? So we're going to use mils of O2 per, and by standard it's per deciliter of blood, which is 100 mils of blood. And the values for this, for normal hemoglobin, or normal blood sample I should say, are 5, 10, 15, 20. So 20 is the maximum, in other words, saturated blood, saturated, right? That's going to be saturated, it's plateauing there. And so saturated normal blood carries 20 milliliters of O2 per 100 mils of blood. And now we can see, we can actually put values on the amount of oxygen that's unloaded, right? Instead of just saying it's a percent desaturation, right? When we go down to, our resting tissues, we're going down to 15%, 15 mils O2 per 100 ml of blood, which means you fell by 5. The 5 you fell by was the amount you unloaded. So now we actually have an amount of oxygen that's being unloaded. Now you go to the VO2 equation, right, and multiply by cardiac output, right, and you can tell exactly how much oxygen is being consumed by the tissues. Okay. Now, getting back to the whole capacity thing, like, okay, this is the capacity of this hemoglobin sample. However, we could have a different hemoglobin sample that gives us, and we're going to assume the hemoglobin is the same. I'm not trying to have a curve shifted in any way. It's not supposed to be different here. Except, it's going to, at 100% saturation, 100% saturated, 100% saturated, right? It's going to carry a quarter less, right, than the normal, what we'll call normal blood, right? We could have another scenario. And again, separate my lines a little bit so we're not right on top of each other. It goes up like that, right? So we can shift down or up in capacity. And we're doing that, so think about that. So the amount of oxygen that's being carried. Well, how is most oxygen being carried? Right, 98.5% of it is carried down to hemoglobin. So to have, to go up to 25 or to fall to 15 means you have to be changing the number of binding sites for oxygen. You have to be changing the amount of hemoglobin. So basically what we do to bring ourselves down, again, not intentionally, but is anemia diseases, right? Diseases that cause a reduction in red blood cell numbers and or the amount of hemoglobin that they contain, right? But it has to include a drop in the hemoglobin content. And going up, right, we basically, if we increase our hematocrit, if we add EPO, erythropoietin, right, the hormone that stimulates erythropoiesis and the production of hemoglobin, now we have a blood sample that has more hemoglobin than typical in it, and so it can carry even more oxygen per unit volume. Okay? Again, you would normally go up in response to erythropoietin. Well, erythropoietin is stimulated by, guess what? Hypoxia. Right? So you become hypoxic, you go to high altitude. Right? Over time, not instantaneously, but over time, you're going to produce more red blood cells over the course of several weeks and end up with a higher carrying capacity. Again, helping to avoid you being hypoxic in a low oxygen environment. When you fall, right, we've got diseases that cause us to lose red blood cells or not produce them as quickly. And again, anemias are one where there's a whole variety of them, but you end up in that scenario where you have less hemoglobin and or fewer red blood cells carrying that hemoglobin circulating in the body. Okay, and again, so that's the third parameter, capacity. So we reviewed cooperativity. That's characterized by the steepness of the curve, right? O2 affecting O2 binding. We have affinity, not shown here. That would be these curves shifting right or shifting left based upon temperature or the allosteric modulators, H plus, CO2, and BPG, right? And then capacity, right? Altering the amount of hemoglobin in our blood sample.